This video I'm talking briefly about just sort of an introduction to the 4 stages, but I'm going to talk mostly about the first stage of mitosis. There are 4 main stages of mitosis, and these are prophase, metaphase, anaphase, and telophase. My students will refer to these as PMAT, sometimes IPMAT for interphase. Okay, so this video we're going to talk mostly about that first stage, prophase. Prophase, okay, obviously starts with that prefix pro. Think prologue, right? Something that happens at the beginning. Prophase is when a whole bunch of stuff happens, actually. The list of what happens in prophase is relatively long. The genetic material inside the nucleus starts as chromatin, right, because we just came from interphase, but then it condenses, and then we can actually see duplicated chromosomes. So notice in our image here, we now have those X-shaped duplicated chromosomes, sister chromatids attached at centromeres, etc. So again, duplicated chromosomes are attached at centromere. The cell starts to build what we call the spindle or spindle fibers. This is basically a system of microtubules. This should remind you of the cytoskeleton. So we actually didn't talk much about the cytoskeleton back in organelles, but this is a huge deal about the cytoskeleton. And these extend from the centrioles, again, something that we listed as a structure inside of cells. It is actually only in animals. Okay, and they are part of the cytoskeleton, and they will be sort of the central system here from which the microtubules in the spindle extend. So here in our picture, these are centrioles. We have 2 of them there, creating a centrosome, and we have these spindle fibers reaching out. Okay, and the centrioles start to move to opposite ends of the pole of the cell. Sorry, different poles of the cell. And the nucleolus disappears, so that little teeny spot that we see inside of the nucleus disappears, and the nuclear envelope ultimately starts breaking down. So here we can see sort of an early prophase. You can see that the nuclear envelope is not nearly as clear as it is in the interphase cells. And then in late prophase, you can see very clearly these linear chromosomes, and they are not in sort of a perfectly round nucleus shape. This blotchiness in the early prophase one is where we are starting to get some of that condensing.